Hi, I'm Cassidy Van Diepstraten, and this summer I had the opportunity to work with the Department of Biomedical Sciences looking at the effects of BPA, BPS, and BPF on bovine sperm fertilizing capacity. Our lab focuses on reproductive health, and there have been a notable decline in fertility levels in humans and animals due to environmental and genetic factors which researchers are just beginning to understand. Bisphenol A, known as BPA, is a common chemical found in plastics such as pop cans, receipts, and toys. It is defined as an endocrine disrupting compound due to its ability to mimic estrogen. In vitro, it has been found to cause abnormal enzyme activity in mature sperm, while in vivo it has been shown to cause DNA damage, impaired motility, and more. However, these are just a few examples. As a result of these studies on BPA, companies use bisphenols with similar chemical structure to BPA, most frequently bisphenol F, known as BPF, and bisphenol S, known as BPF, that are the most commonly used, safer alternatives. However, there's a lack of evidence supporting this. In the United States, 92.6% of urine samples contained BPA, while 78% of samples contained BPS and 55% contained BPF. Long-term exposure to bisphenols has been shown to cause epigenetic changes, with these changes in gene expression being heritable without the alteration to the DNA itself. In the past, our lab has looked at microRNA expression, a small non-coding RNA that post-transcriptionally regulates gene expression. These miRNAs have been used as diagnostic biomarkers to assess male infertility and to analyze gene expression. For the proposed methods, we would use the lowest observable effect level for BPA, which is 0.05 milligrams per milliliter. We are interested in using ELISA to determine the persistence of BPA, BPS, and BPF in both sperm and seminal plasma. An additional experiment would be conducted on sperms using spermac stain. The 3 stains would be used to clearly identify the different components of the sperm. This would allow for the identification of morphological defects and what are the most commonly found. We would then use a grading system to determine their progressive motility, the strength and speed of their swim, and their swim pattern. From this point, we would extract the RNA from the sperm and reversibly transcribe it into cDNA using a poly-A polymerase and a Q-script reverse transcriptase that would then be analyzed by qPCR. This procedure could then be followed for seminal plasma or embryos to detect, characterize, and quantify mRNA in samples. To determine the sperm's fertilization capacity, we would use the sperm that was stained to fertilize the oocytes. We would then collect oocytes at the 2 to 4 cell, 8 to 16 cell, and blastocyst stage, and then follow the same procedure to conduct a qPCR. Lastly, we would use seminal plasma to analyze protein expression by completing a protein extraction and then a Western blot. For the purpose of this study, we will determine how the LOAEL dose of BPA will affect the sperm's molecular mechanisms and its ability to successfully fertilize an oocyte. We are also hoping to identify specific proteins or miRNAs in bovine that demonstrate infertility caused by bisphenols, with the possibility of identifying a biomarker. We are looking in to see how gene expression could be altered in vitro after exposure to treatment with BPA, BPS, and BPF in the sperm, seminal plasma, and embryos. We are hoping to see a difference in protein and miRNA expression in the sperm that have been exposed to treatment groups in comparison to the control group. To conclude, this study aims to present novel findings of how BPA, BPS, and BPF alters expression in mature bovine sperm and seminal plasma, and how fertilization and gene expression in embryos are impacted by these chemicals, with the hopes of translating this research into how humans and other animals are reproductively impacted by BPA, BPS, and BPF. I would like to take the time to thank my faculty advisor, Dr. Laura Favetta, PhD students Reem Sabri and Ola Davis, and our lab technician, Liz St. John. Thank you to the University of Guelph's Biomedical Science Department and NSERC for funding this project.